Good afternoon, everyone. Welcome back to the Harmful Algal Bloom Research Symposium. The Algal Bloom Action Team is thrilled to be able to host this event for a fourth year, and we are glad that you can join us. This year's symposium is organized into two sessions. If you were here this morning, the morning session had three presentations focused on HAPS monitoring and treatment. There was also a special feedback session related to lower ocean acidification program. In this afternoon, we will focus on HAP outreach and communication. I'm the moderator for this afternoon session as a member of this Algal Bloom Action Team. My name is Baoning Deng. I'm a professor in the University of Missouri Columbia campus. I'm a professor in the Civil and Environmental Engineering Department. I co-direct Missouri Water Center. My research is focused on aquatic chemistry, water quality assessment and monitoring, as well as water treatment and reuse. Next slide, please. A few words on the Algal Bloom Action Team. This team is a collaboration of water professionals, researchers, and educators from 12 states in the North Central region of the United States. Team members include the National Network of Water Resources Research Institutes, the North Central Region Water Network, and the University Extension within each state in the North Central region. In addition to hosting this annual symposium, our team is developing a website of resources that can be used freely for outreach and educational activities. I encourage you to visit our website, that is northcentralwater.org/HAPS. I will try to put this link to the chat later on. You can explore some of the resources we have put together that include what you should know fact sheet, a fact sheet on preventing and treating HAPS for non-owners, and the HAPS frequently asked questions database. We're also excited to announce that today we are releasing a fact sheet focused on HAPS and their health-related effects on animals. You can find that fact sheet and others at our website. In addition to this symposium, we also host a bi-monthly webinar series where we feature the latest HAPS research. On our website, you can find the recordings for our past webinars, information on upcoming webinars, as well as access information and recordings on our past HAP research symposiums. Next slide, please. Before we get started, I just want to have some housekeeping items here. We will have two 20-minute presentations this afternoon. Followed by each presentation, there will be 10 minutes of Q&A. We will allow time for Q&A after each presentation. Please post your questions using the Q&A panel, which can be found at the bottom of your Zoom screen. If there is a question already posted to the Q&A panel and you have the same question, please use the Zoom app vote function on the similar questions to help ensure we get to answer your questions there. Please note the Q&A panel function is separate from the chat function in Zoom. Again, questions for the presenters should be posted to the Q&A panel. If you are having any technical difficulties during the symposium, please let us know using Zoom's chat function, and we will happy to assist you as best as we can. You can also use the chat functions to introduce yourself or discuss the research findings with fellow water professionals at this symposium today. All symposium presentations are being recorded and will be posted later to the Alga Bloom Action Team website at the conclusion of the event. We also posted the presentation slides to this website for your reference. Next slide, please. Without further ado, let's just get started with this afternoon session. The first speaker we have is Lindy Holt. Lindy received a degree in biology, environmental science, and chemistry from the University of Derbyshire in 2021, where she was engaged in climate change research focused on measuring ecological impacts occurring on the Mississippi River. From there, she went on to graduate school where she received just recently, 2023, with a master's degree in occupational and environmental health from the College of Public Health at the University of Iowa. Now she worked in the human toxicology and exosomics laboratory. Great to know this new word here. A lot of omics under Dr. Peter Thorne, where they were a part of the multidisciplinary Iowa Healthy Lakes initiative, studying human exposure to cytotoxins and Iowa's understanding, attitude, and information reception as related to harmful algal blooms. This work is being continued at the Iowa Lakeside Laboratory in the Iowa Great Lakes region, where she is currently serving a term as an environmental steward through the Green Iowa AmeriCorps program. Without further ado, let's welcome Lindy for her presentation.
All right, thank you. Share my screen quick. All right.
Yeah.
Does that look good?
Looks good.
Okay, thank you. All right, so I'll jump right in as to not run into the next presenter's time. This work was a part of my master's degree at the College of Public Health at the University of Iowa. Quick agenda, I'm going to talk about the project that this work was a part of, and then 2 parts to this project was a survey and then a hazard assessment, and then we'll talk some recommendations at the end based on this work. So during my time in my master's program, I was a part of the Iowa Healthy Lakes Initiative, which was a multidisciplinary team of researchers from all across campus coming together to address the problem of harmful algal blooms in Iowa lakes, taking kind of a holistic One Health approach to it. And so I was a part of the Public Health Communications and Community Engagement Working Group. And we had kind of 2 aims with this group. We were focused on investigating potential gaps in public health protection, and then understanding kind of the perception of harmful algal blooms in Iowa, specifically to Iowa residents. And so that kind of included looking at what people are being potentially exposed to at Iowa beaches, what cyanotoxins are present, in what concentrations, and then what kind of recreational behaviors are associated with this. And then also understanding in a broad sense, kind of like, what do people know about harmful algal blooms? How are people getting this information? How do they want to get this information? What kind of information do we need to push? Things like that. So the first part of this was a survey. It was called the Iowa Water Recreation Survey that we developed. And right here is a picture of the physical flyers that I handed out during the time that this survey was open, which was from about March 17th, 2023 until Labor Day 2023. So over the period of a couple of months, disseminated this survey within kind of my own networks and groups through mass emails with the university, physical flyers that I had posted at several locations, at the Lakeside Laboratory. I kept flyers on hand to hand out at any events, like such. I was at Hawkeye Caucus Day at the Capitol, had it posted at breweries or any other water quality type events that I was attending, as you can kind of see in that picture there. Shared it on all of my social medias, where it was promoted by various groups like conservation groups, different nonprofits who also shared it in like their newsletters, email blasts, those kind of things. So I had a lot of help disseminating this survey, and I ended up getting over 1,900 responses. Only 1,655 people actually completed the survey, but over 1,900 did answer at least one question, which was pretty cool, way over the goal that we had set for this. And then I did have a friend also post it on an Iowa subreddit, which is that picture you can kind of see at the bottom. I just included that because I thought it was kind of a comical hill to die on. Won't promote anyone attending the University of Iowa. And so the demographic distribution of respondents for this survey was concentrated pretty much in the urban centers of Iowa, definitely Johnson County, Polk County, Linn County, and then Scott and Dubuque County. And then we've kind of spreads out from there. And then we've got a little bit of a hotspot up in Dickinson, which is where the Iowa Lakeside Laboratory is located. And so based on those demographics, they were compared to the state of Iowa demographics just to see how similar they were. And they were similar on quite a few categories. They weren't able to be compared on all categories. For the p-values that are less than 0.001, those comparisons are statistically significant. There's not a significant difference between those. And so I like to say that our typical respondent was Dr. Barbie, because a respondent was most likely to be white, female, working age, have an advanced degree in healthcare and work in healthcare. Overwhelmingly, people had an advanced degree that took this survey, which definitely skews it. But I think that kind of just reflects the network and where it was pushed out to, especially in Johnson County, going out through the university. Many people with an advanced degree probably ended up taking this survey more than usual. And so the first part of this survey was asking about recreation. We just wanted to know, where are people recreating? How are they recreating? And this also reflects the demographics. By far, the most visited was Lake McBride, which is located in Johnson County. And then you also have, Backbone, Big Creek, also big spots for recreation in the state of Iowa with beaches. And how are people recreating? Most people were participating in non-wildlife recreation, and the most chosen pastime was swimming. So people are engaging in non-wildlife recreation, some people doing both, but most of the time they're in or near water, doing things like swimming, kayaking, boating, even hiking and dog walking includes being, near water many of the times. And then people were asked if they leave Iowa for their water recreation. And that was a pretty split bag. Some people said yes, some people said sometimes, some people said no. After that, people were asked if they would use Iowa's water resources more if there was better water quality. And people overwhelmingly said yes for this, definitely, almost 80%. And this question led into the next question. Do you have concerns about the water quality in Iowa? Over 1,700 people answered this question, and 3/4 of them said yes, which is a pretty startling amount. A couple people were still unsure, but there were not many straight no's. And so after being asked about recreation, then we kind of got into the harmful algal blooms. Most people have heard of HABs in the state of Iowa, at least the ones that took this survey. But then when they were asked what they look like or if they could identify them, that number dropped pretty significantly to about 52%. Next, people were presented with the picture on the left there and asked, have you seen something like this in Iowa? And then that number dropped even a little bit more to 50%, which kind of reflects what I've seen in my field work is that people see anything green in the water and assume that it could be hazardous. And that includes, non-harmful filamentous green algae or duckweed, things like that. But then people were asked if they are aware of the adverse health effects that can be associated with exposure to harmful algal blooms and cyanotoxins. And the number jumped a little bit to 55%. So a majority of people know about them, aware of the health effects, and have potentially seen one. Many people were still unsure about that. And so next we wanted to know, how do you know this about harmful algal blooms? Where are you getting your recreational information from? And people were asked to select all that apply to this question, and these were the options. But most people are using the Iowa Department of Natural Resources. I'll go to the next slide, click. This is, on the left here is a screen cap from the DNR's beach monitoring site. It's going to look a little bit different after this year, I believe, but this is from 2023. It has all of the state park beaches listed on a map. With it's color coded, so at first glance you can see where swimming is recommended and where it's not, and then once you click on a site, you can get more information like concentrations on E. coli and microcystin, which is what the DNR tests for weekly during the summer in Iowa. But the IEC, the Iowa Environmental Council, also does the same thing. They have a map with all of the state park locations and their results. And these two sites also include results from the U.S. Army Corps of Engineers. Their results from beach monitoring, they cover reservoirs in Iowa, such as Coralville Reservoir, Saylorville Lake, places like that. And so back to recreational info. So most people are using DNR. Other than that, people said that they use social media, various different groups, social media, and some people even said none. They don't have, they don't use anything. And so with the big three, we wanted to know specifically what resources they're using. And it's pretty much just websites. People want to check a website that's consistent, can be a lot more consistent than social media. Other than the website and social media, Iowa Environmental Council sends out a weekly Water Watch newsletter on Fridays every week during the summer, and a small amount of people were using that also. For the other options that people selected, we prompted them, what do you mean by that? What groups, social media, what community organizations, what does other mean to you? And mostly people just said Facebook. Whether that was county conservation, city pages, parks and recreation, different kind of recreational groups, such as like rowing clubs, like Iowa Water Trails, paddling clubs, swim clubs. The cow icon on the right is for the Iowa City open water swimmers. Obviously, you need to kind of be in tune with what's going on in the water if you're going to be actively recreating in it. And so with all this information, we asked people, how do you want to know about this stuff? What's the best way to reach you? And overwhelmingly, people just said, check a website. So what they're already doing is what they want to do. And that makes sense. It's consistent. You have it bookmarked, you check it before you go, okay, I have the results. But people also wanted physical postings still. And then the next highest was social media. And so we'll talk about this a little bit at the end with our recommendations, but what this kind of showed us was having kind of like a safety net. So you have a website for people that are looking. Maybe you get some more eyes in the form of social media. Everyone seems to get on that every single day. And then your last resort is you have that physical posting at a beach site. And so, you can ignore maybe the first two, but the third one you can't. because all we really want to do in terms of public health protection is get this information in front of people's eyes so that they can make the best informed decision for their health. And then also there were several answers for other, and when prompted about that, most of them called on organizations like local news or Iowa Public Radio to talk more about this. A lot of people listen to Iowa Public Radio on their commutes or during their lunch breaks, things like that. And so lastly on the survey, we asked if it changed harmful algal blooms, the presence of changes the way people recreate. And this question came out of some interviews I did with DNR employees who said that there's three kinds of people when it comes to harmful algal blooms and advisories. There's people who will actively avoid the site, actively avoid the water, or just not put their head underwater while swimming. Thankfully, we didn't have very many people that said that that's all they're doing. But for the most part, people said, yeah, this will change the way I recreate. It could determine whether I, keep visiting or it depends. Many people said it depends, which is good. We don't want people to go about their business when there's a giant orange warning sign in front of them. And so along with this, we wanted to look at what kind of toxins that we're seeing that people might be exposed to in Iowa. And so we did a bit of a hazard assessment. These are our sites kind of based in Northwest and Southeast Iowa. Not all of them were state park beaches. Here's a slide on methodology, which I believe these slides will be available for you to look at. I adapted my method from the EPA's method 546. And so we looked at microcystin, which is the only one the DNR tests for. The advisory is issued at 8 micrograms per liter. When a sample crosses that is when they'll issue a swimming not recommended advisory for a beach. And we had 4 samples above this this advisory level. And so these results are that you're viewing right now are log transformed, but the line is still for where the advisory limit would be. So we had four above that, as high as almost 37 micrograms per liter. So well above that advisory limit. And interestingly, our highest sample was not at a swimming area or a beach area. It was at a popular fishing area at Lake McBride in Johnson County. But this is an area where open water swimmers often take off from, that people are still being exposed to, especially if you're fishing, you splash by water, maybe get a little bit ingested or something, can still cause harmful effects. And then here's those results again, separated into regions. So Southeast was the one that had all of the samples above this advisory limit, and 3 of those are Lake Darling, which if you know anything about Iowa lakes, Lake Darling's a kind of a problematic one these days. And so we also looked at anatoxin, which is not something that's often tested for in Iowa. And anatoxin is a neurotoxin. And so we only had 2 results above that advisory limit, and there is no federally set advisory limit. And so I pulled 2 from both the state of Washington and Minnesota. But for the purposes of this graph, went with the Washington 1 microgram per liter advisory limit. So only 2 above that, but the results were pretty startling. One sample, we found 248 micrograms per liter of anatoxin in a sample that did not test high for microcystin at all. Conversely, the highest sample from the microcystin bunch was also high for anatoxin at English Point. Anatoxin and microcystin were found in similar concentrations. So here's those results again. Again, you're viewing these as log transform values, just to get the whole spectrum, because we had everything from below the limit of detection on our assay to well above that advisory limit, well above detection limit, where we had to dilute and rerun these a couple of times using the ELISA method of detection. And we also tested for saxitoxin, which is typically thought of as a marine toxin, often thought of as paralytic shellfish poisoning. So not much to say there. The advisory level is about 30 micrograms. We were nowhere near that. So in this case, no news is good news. But we were not expecting to see much on that end. And so to touch quickly on recommendations, and to conclude, we came up with a couple of things based on what we found here. First, increasing the testing suite. As we saw with the high anatoxin sample, there was no indication from the microcystin levels that this would be a problematic beach. So doing further research to identify areas that are going to be more prone to different kinds of blooms such as this, and then testing those based on those results, rather than testing for something that we're not going to find there, and issuing advisories based on that. That's, you know, a gap in public health protection, especially with a neurotoxin. And then next, the use or development of a reporting website. We don't want to reinvent the wheel here. There are certainly already applications that do this. I believe Bloomwatch is one. But this could help alleviate some of the burden that state officials already have, especially for local areas where they're not testing. They can't have eyes everywhere at all times, but by kind of providing this citizen science funded project, you'll be able, people are still getting the information they need to make the best decisions for their health, and kind of giving people the tools to make the differentiation between harmful and not harmful. And yeah, many people are going to the same places consistently in the summer, especially, you know, maybe to walk or walk their dogs. Why not utilize them if they're there and willing? And then also doing an evaluation of signage. I didn't touch on this much with the discussion of the project, but the language on the signage on the left there is a sign that is issued when there's an advisory at a beach in Iowa. Very small words. It's orange, which is, you know, a attention-seeking color, but the words are very small, and that's really all the information you get. They do list the DNR website on there, but also it's only in English. We have quite a large Hispanic population in Iowa, especially that could utilize these beaches in the summer. And I compared it to a picture on the right, which is actually from Wisconsin, and we'll hear from Wisconsin in a couple of minutes. But I really like what they did is they gave people pictures of the harmful algal blooms and the not harmful algal blooms, which is just another tool to give people to make their own informed decisions. And then their sign just says water quality today is good and it's green. And I think that's great. The simpler you make it, the better. And when the water quality is bad, I'm assuming the sign is red. Very simple, gets the point across. And then having the availability of this information through multiple channels. We already talked about this a little bit, but having multiple safety nets of like, well, if we don't get them here, we'll get them here. Okay, we're bottom line, we're going to get them when they get to the beach, which we're trying to avoid. We don't want people to drive 2 hours to a beach to find that it's closed, but that's better than nothing at all. And then lastly, an increase in funding or an expansion of programs. The surrounding Midwestern states have really great examples of programs, one that we'll hear from next, that I think could really be integrated into Iowa's process. Especially like stuff like this where it's dynamic signage, and just having more room for flexibility within our own program. And so I know I'm running out of time here, just a couple limitations. First time I've done a huge survey, definitely would, if I could go back and do it again, make some changes with wording and ordering of questions. Some of them were leading questions. Some of them people were confused by, and some of the feedback that I got. And then for the exposure assessment, we just did a single snapshot. Would have loved to do more of a time series, but currently this work is being continued at the Iowa Lakeside Laboratory, and doing making some of those corrections that I just touched on in kind of a larger project there. So that's it. Thank you.
Thank you for the nice presentation there. We got quite a few questions for you there. Let me see. We'll try to let you to answer as many as possible. The first question is that for your survey, did you provide a definition of HABs? That is by Sarah Davis.
No, so we just asked people, have you ever heard of harmful algal blooms, as like the first question, and then got more into detail with those questions as the survey went on. So have you seen one, or do you know what it is? And then we gave them the picture, and then asked about any adverse effects. But other than that, we did not define it for them completely.
Okay, thank you. Another question that quite a few asked is you mentioned that you could reach people who are well educated, but do you have a way, sort of, what kind of approaches you may use to reach out a broader, less educated audience?
Yeah, so that's something that we want to focus on this summer, because we were more concerned, especially with completing the master's and just getting a certain number of responses, and ended up being very skewed. And so as we kind of continue this research and do more, we're looking at more ways to target more of a normal population. One of the ways that we did that just didn't end up working out is I put my survey flyers into these free boat bags that we gave out all summer. Up at Lakeside, we would go to different beaches and hand out these free bags to boaters. But right now, yeah, we're coming up with a lot of different ways that we can target more of a diverse population. And I think that's going to include me just going to beaches in the summer and asking, with an iPad, and asking people if they'll participate in a survey.
Yeah, a related question I have is that, have you ever thought that just go to the community, try to find the community leaders or people who are interested in the issue, just try to talk to them?
Mm-hmm. Yeah, I think that's a great idea. It was just tough with trying to get the survey out to finish my master's on time. And then also much of this work in the summer was done in the Iowa Great Lakes region, which is a really tourist heavy destination. And so a lot of those people were not from Iowa. So that made it a little bit more difficult, where we're hoping this summer that we can kind of focus in on a community. And we were thinking more of like the Storm Lake community, because that's a much more diverse population.
Okay, great. The next question is from Melinda Cutler. The question is, how frequently are cytotoxin samples collected from the sites? Are there any other parameters used to screen for likely HABs before testing for cytotoxins?
So in terms of the Iowa DNR, they test for E. coli, and they test for microcystin once a week at the 39 state park beaches from Memorial Day to Labor Day. For this work, for my results, we did a single snapshot of a week, took multiple samples from several beaches, and that is the results I showed here. So not a great reflection, but more of just a snapshot in time. Other states, I believe, use different measures to instead of just going straight to toxin testing, but that's what the IDNR does.
Okay, thank you. The next question is by Hannah. Were the main cyanobacterial organisms identified in the anatoxin A and microcystin toxin analysis?
Cyanobacterial.
Specific, what kind of the main cyanobacterial organisms? I think it's talking about specific species.
I did not do species identification. I think that would be a great next step, especially as we see, some places are prone to anatoxins, some are prone to both. What species are we seeing in those areas? But I did not do any species identification for this.
Okay. The next question is still on this survey. It's by Oder Nake. I don't know whether I pronounced the name correctly. In this survey, did you ask whether respondents know some of the factors that contribute to HABs?
I did not. All we really asked about after we asked about recreation was just, do you have concerns about water quality? And the other question of would you use it, the water resources more if water quality was better? But yeah, we didn't ask anything specific about nutrient pollution or climate change or anything like that.
Okay. Now the next question is by Steve. The question is, can you make ALS in humans or animals due to blue-green algae a reportable disease in Iowa in order to better track these cases?
So microcystin-related illness is a mandatory reportable illness in Iowa. The problem is especially with adults, it's not really for a normal healthy adult that might ingest some microcystin from the water. Many people are going to chalk it up to, well, I was in the sun all day, or I was drinking and I'm hungover, or I'm dehydrated. When really you might be experiencing microcystin-related illness symptoms. So many people aren't going to go to the doctor for that. They're not going to report it. The cases that we do see being reported are usually when people are very sick or when vulnerable populations are exposed, such as children or dogs. Those often experience much more severe symptoms, especially in dogs. But just to touch back on that last question about asking about factors and stuff, there's quite a few surveys in Iowa that already ask about that, that were used kind of as comparison with this project. But we wanted to specifically just ask what people knew about harmful algal blooms.
The last question I have here on this Q&A panel is that as climate is changing, do you expect HABs to be more frequent in Iowa?
Oh, absolutely. Definitely had to cut this presentation down just because of the time. But yeah, we're seeing increased rainfall in certain times of the year in Iowa, and then of course an increase in temperatures. But yeah, with this increase in rainfall, we're having more nutrient pollution combined with increasing temperatures. It's kind of the perfect breeding ground for harmful algal blooms in Iowa lakes. And so I imagine that it's only going to increase in frequency and severity, and also variability, especially with as our weather kind of increases in unpredictability. So will environmental concerns such as this.
Well, thank you again, Lindy, for the wonderful talk. And the time is perfect. We just try to move on to the next speaker.
Thank you.
The next talk is by Jordan Murray. Jordan is a harmful algal bloom epidemiologist stationed in the at the Wisconsin Department of Health Services in the Division of Public Health, where Jordan is managing the harmful algal bloom program. Her work entails prevention and control of harmful algal bloom related illness through epidemiological research, case investigation, and outreach. She holds a BA in neuroscience and a master's degree in public health epidemiology. Jordan, all yours.
Thank you. All right, yeah, thanks so much for that introduction. I'm just going to turn my video off just for the purposes of the presentation. I'll turn it back on after the presentation's over, just because my Wi-Fi is a little spotty. But yeah, thanks for that introduction. Today I'll be talking about the health effects associated with exposure to cyanobacteria and cyanotoxins and how our HAB program at the Wisconsin State Health Department deals with these illnesses. So it is not really question that cyanobacteria can be a health hazard. Cyanotoxins can make people and animals sick if they are exposed to them. The signs and symptoms that develop from exposure to cyanobacteria and cyanotoxins depend mainly on things like the route of exposure, the species of cyanobacteria that we're dealing with, and what types of toxins they produce, as well as the concentrations of the cyanobacterial cells and toxins, and also existing vulnerabilities. So certain factors like behavior, body size, pre-existing conditions like skin sensitivities or liver disease can put a human or animal at an increased risk of not only becoming exposed, but becoming sick after the exposure occurs. People and animals can be exposed to toxins through a few different ways. So ingesting or swallowing water is the most likely exposure route, especially for children. Swallowing water with toxins in it can lead to gastrointestinal symptoms like cramps, nausea, diarrhea, and vomiting. Ingestion of neurotoxins can cause symptoms like dizziness, numb lips, and tingling fingers and toes. In Wisconsin, we don't typically see neurotoxic effects, but when we do, they're most commonly observed in dogs. And skin contact is another possible exposure route. So if a person comes into contact with algal scum or mats, they might develop a rash, hives, or skin blisters. I do want to point out, though, that toxins aren't absorbed through the skin and into the body, so we wouldn't really expect them to cause symptoms beyond skin irritation. The last exposure route I want to mention is inhalation. So when people breathe in aerosols that contain cyanotoxins, they might experience cold-like symptoms like a runny nose or eyes, sore throat, or a cough. There isn't a ton of information on this exposure route just because it's quite honestly just hard to study, but it's definitely a pretty hot topic in the HABS world right now, and I'll touch a little more on it later in the presentation. And then switching gears to animals, sadly, they are affected by cyanotoxins too. We've seen adverse health effects and even death in dogs, wildlife, and livestock who are exposed to toxins. They're especially susceptible because, unlike humans, they're not as easily deterred from the smelly green water. They also tend to be smaller in size, meaning that it doesn't really take much toxin to make them sick. And because they need a relatively smaller dose to become ill, severe symptoms are more commonly seen in them. So symptoms can include things like neurotoxic effects like lethargy, muscle weakness, paralysis, seizures. We might see gastrointestinal symptoms like vomiting and diarrhea. Respiratory distress can also occur, and the animal might have difficulty breathing. Because toxins are fast-acting agents, the onset of symptoms in animals and humans as well generally occurs within minutes to hours following exposure. So the symptoms I just explained will typically happen with acute exposure to HABs. On the other hand, the health effects of chronic exposure to HABs are not as known. However, some animal studies suggest that chronic low-dose exposure to cyanotoxins may be a concern for people with pre-existing conditions, and exposure over time can potentially exacerbate these conditions like non-alcoholic fatty liver disease or liver cancer or airway and lung inflammation. But again, not much research has been done regarding this topic. Moving on, I'm going to talk a little bit about the Harmful Algal Blooms program at the Wisconsin Department of Health Services and the work that we do. Our goal is pretty simple. Since 2008, we have worked to prevent and manage HAB-related illnesses, and we do this through illness surveillance, outreach, and research. For 15 years now, we've worked with state partners to investigate HAB-related illnesses in Wisconsin. This partnership consists of us, the Department of Health Services, the Department of Natural Resources, and the State Laboratory of Hygiene. And just putting some faces to names, here you can see 3 groups who make these partnerships possible. So my former mentor, she was my mentor for 2 years. Her name's Amanda. She and I are up top here posing with the bloom. On the bottom left is Gina LaLiberty. She's our statewide harmful algal blooms coordinator at the Wisconsin DNR. And on the bottom right is Dawn Perkins. She is our state lab microbiologist. And to summarize what our program does, we conduct surveillance of the health effects related to HAB exposure. We investigate reports of human and animal illnesses. We coordinate water sampling and analysis. We help local public health issue health advisories and beach closures, and we provide education and outreach. So now I'll talk about how those activities fit into a typical illness investigation. First, the DHS will receive an animal or human illness complaint. We most commonly receive these complaints through our website, but we also receive complaints via phone calls or emails from the complainants, or through referrals from the DNR, local health departments, lake association groups, the Wisconsin Poison Center, and sometimes even clinicians. Here's a screenshot of our DHS HAB websites, and you'll see at the bottom of the page here is that link that I mentioned to report a suspected HAB illness. So after our program receives the health complaint, we begin the investigation by interviewing the patient or animal owner. We ask a series of in-depth questions about the nature of exposure, symptoms, and health history. Our goal is to figure out if the illness in question could be from exposure to cyanobacteria or cyanotoxins, or if another cause is more likely. More oftentimes than not, when we receive these illnesses, we end up determining that the illness in question is actually not related to cyanobacteria. So that's kind of just why our questionnaires are so thorough. But in the end, if the illness sounds like it could be due to cyanobacteria, we'll usually contact Gina and the local health officer to inform them about this illness. Then we'll decide if water sampling is necessary. So to do this, we call up Gina to gauge her thoughts about whether sampling the water is justified or not. What we want to know is basically, is the water representative of environmental conditions at the time of the exposure? So what does the water look like now? How many days have passed since the person or animal was exposed? Or have significant environmental events caused, or are they suspected to cause changes to the bloom before sampling? So if the illness in question sounds like it could be due to cyanobacteria, and if water conditions are still representative of those at the time of exposure, Gina will then work with regional DNR staff to collect water samples. This protocol does instruct field staff to wear rubber gloves during sample collection, so we recommend that people don't do this because that's how you get rashes. Samples are then sent to Dawn at the State Lab of Hygiene, and Dawn will then microscopically identify and enumerate cyanobacteria in the sample, as well as conduct toxin testing. After Dawn analyzes these samples, she'll report the results back to me and Gina. And then finally, based on the results, we'll interpret the level of health risk present and take the steps necessary to protect public health. We typically share the water sample results and the health risks with local health officials. Wisconsin is a home rule state, so local health has the authority and the responsibility to post beach closures, health advisories, or just generally speaking, restricting access to a swim area when a bloom is present. But even though local health has this responsibility, the DHS and DNR will often provide them guidance and recommendations for what sort of public health action could or should be taken in the event of a bloom or illness. Wisconsin doesn't have standardized recreational guidance for exposure to cyanobacteria or cyanotoxins, so we often defer to federal guidance when interpreting test results. Listed here are some guidelines from the Environmental Protection Agency and World Health Organization for cyanotoxins. The DHS recommends that local health issue a beach closure when testing of cyanotoxins or visual observation of a public swim area suggests a high probability of adverse human health effects. Now I want to talk about a case study related to a HAB outbreak that our program investigated pretty recently, about a year and a half ago. Back in June of 2022, right before the 4th of July weekend, of course, we received an illness report through our online reporting tool regarding 3 human illnesses. So 3 recreational camp staff members became sick with respiratory and dermal illnesses after swimming for about 15 minutes in the camp's private swim area. We delivered our standardized interview to 2 of the 3 illness complainants, and during the interviews, the camp staff members described the water in the swim area at the site and during the time of the exposure to be green with a thick layer of little floating blobs, and that you couldn't really see your feet in the water. This is pretty consistent with what we think of when we think of HAB conditions. We also learned that camp staff member A became sick about 3 hours following exposure. She experienced a headache, eye irritation, and congestion. She has a history of autoimmune disease and seasonal allergies. This information sounded pretty consistent with a HAB-related illness, so we ended up classifying it as a probable case. We learned that camp staff member B became sick about 3.5 hours following exposure. She experienced a headache, abdominal pain, eye irritation, congestion, and a rash under both of her eyes. She has a history of eczema and seasonal allergies. And as mentioned earlier, people with skin sensitivities are typically more prone to developing dermal irritation from exposure to a toxin. Again, this sounded like it could be related to HABs, so we classified this illness as a probable case as well. And then we learned indirectly from one of these 2 complainants that the 3rd illness complainant developed pretty severe allergy symptoms and actually had to visit an urgent care. Unfortunately, we were unable to get in touch with this person to learn more about his exposure and illness, and because we had almost no information, we had to exclude him from our investigation. While we were working on coordinating the water sampling, the camp staff had informed us that there is a very busy public beach next door to the camp, located just a little downstream, that was also experiencing HAB conditions. So what did we do? Well, we notified the local health officers of the respective jurisdictions, and we collected and sent water samples to the State Lab of Hygiene to be tested, analyzed, and interpreted for public health purposes. The cyanobacteria identified in the water sample were Microcystis aeruginosa and Aphanizomenon flossiquae, which are 2 of the most common bloom formers in Wisconsin. Microcystin was detected at a low concentration of 3.2 micrograms per liter, which, if you remember from the slides earlier, is below the EPA's recreational guidelines for cyanotoxins, which is 8 micrograms per liter for microcystin. Now, it did take about a week to collect samples after the exposure, and conditions easily could have changed in that time. So considering this sample may not have been representative of the conditions at the time of exposure, the key takeaway really was that microcystin could be produced by this bloom. So if a bloom is present at the beaches, then swimming may be risky to health. The DHS and DNR worked closely with local officials to provide them guidance on what type of signage would be most beneficial to post at their beach for the water conditions at the time. During the investigation for several weeks, Gina and I would regularly, almost daily, receive photos from the staff at both of these beaches. Shown here are some examples of the pictures they sent. Gina and I would assess these photos and then provide feedback about the level of health risk present and what type of public health action could be implemented based on the conditions portrayed in the photos. And then lastly, we offered public health messaging examples and resources to both the camp staff and local health departments. And what they did was they physically distributed to beachgoers and these flyers, and then they also posted content on their website and social media platforms. So given all of this information, it's pretty easy to see that HABs are an important public health issue, but their health effects are still not fully understood. This gap in knowledge stems partially from the lack of large-scale epidemiological research on this topic. And because of this, we really rely on people reporting cases to us so we can learn more about these illnesses. One of the challenges we face, though, is that these illnesses are not always reported to us. And over the years, we've kind of learned that there are a few reasons for this. So not everyone is aware of what HABs are, what they look like, that they can cause illness, and what kinds of symptoms exposure can produce. Most symptoms of HAB illnesses are so vague that they sound like other conditions like swimmer's itch, food poisoning, or allergies. HAB-related illnesses, they don't have a single signature sign or symptom that can kind of clue us in. HAB-related illnesses are also very challenging to diagnose from a medical standpoint. In most cases, illnesses are not severe enough to warrant medical attention, and even in cases severe enough to be seen by a doctor or veterinarian, the level of awareness of HAB-related illness among medical professionals can vary pretty considerably. And so being seen does not guarantee that that practitioner will recognize the illness as HAB-related. And then on top of that, there's currently no commercially available diagnostic tests that the doctor could run, for example, on a blood sample to confirm that an illness was in fact due to HAB exposure. So essentially, these are just some of the reasons that there might be a potential underestimation of the number of HAB illness cases that we see not only across Wisconsin, but across the country as well. And I do want to give an example of how Wisconsin is addressing some of these public health challenges and how we're taking initiative to better understand these illnesses. So as mentioned earlier, inhalation of cyanotoxins is a pretty understudied topic. We don't really know exactly how significant this exposure route is, how frequently it affects people, and what populations are most susceptible. So for the past 2 years now, Wisconsin has been leading a collaborative project with several other federal agencies like the CDC, US EPA, and NASA on research to help provide clarity and more information on the relationship between cynoHABs and respiratory health. Our region of interest was Green Bay. We compared cyanobacteria data characterized as chlorophyll A values. We compared these data to respiratory-related electronic health records for residents living in the surrounding Green Bay area from 2017 to 2019. Now, we didn't find any significant associations between hospital records and cyanobacteria concentrations, but this doesn't necessarily mean that there isn't a relationship between them. As expected, given the novelty of this study, we had some limitations like small sample size and confounding variables that easily could have hindered our results. But this doesn't mean that our research wasn't important. To our knowledge, this is the first study that has modeled the relationship between HAB occurrence using cyanobacteria satellite data and respiratory health using hospital records at a population level. So that in itself is pretty monumental. And we're also doing this again. So we're currently in the second phase of this project where we're looking at 2 more water body regions in Wisconsin, both of which are pretty infamous for their bloom issues. This is exciting because we'll have a larger sample to work with and we'll have the opportunity to refine and improve our methodology. And then just wrapping things up here, I just want to briefly mention some of the educational resources that our program has created and that we offer to people. So we've created fact sheets as well as signage, and thank you, Lindy, for highlighting some of Wisconsin's signage in your presentation. but we have the signage that can be posted during the swim season. Currently, we're working on a fish fact sheet, fishing and fish consumption fact sheet that will hopefully be available before this year's HAB season. Our most popular resource by far are these educational HAB bookmarks. People of all ages and backgrounds literally go crazy for them. I just ordered a couple thousand more of these. So if anyone is interested in receiving some, please let me know because I have plenty to spare. You can find these resources and more by visiting our website listed here. And that's all I have for today. Thanks so much for listening. I'm happy to answer any questions at this time.
Thank you, Jordan, for the wonderful talk here. It's very informative here. Let me see. There are quite a few questions here. The first one is by Karen. The question is, how many illness complaints do you typically receive in a season? How many of these do you determine are likely HAB illness?
Mm-hmm. Yeah, I actually had a graph that I ended up taking out of this presentation that showed frequency of illness complaints. But to date, from 2009 to 2023, we've had a total of 333 illness complaints reported to our program, with an average of about 25 per year. And out of the 25 per year human or animal illnesses, we only determine maybe a fourth of those illnesses to actually be related to HABs. And even in those cases, we're still not able to completely confirm that these illnesses are related to HABs. We just have to use our best judgment. But yeah, only a very small handful.
To follow up on this, if you compare Wisconsin with other states nearby, they're comparable?
Mm-hmm. Yeah. That's
Missouri.
Yeah, that's a tough question. There, I imagine other states have a similar issue where they're not, illness reports are underreported. And so because of that, people aren't receiving a ton of illness reports, but other states have similar issues where they receive these reports and only a small portion of them are actually determined to be related to HABs. But each state is very different in how they investigate illnesses and how they collect illnesses and how illnesses are reported. But there actually is a national database. It's called OHABS, One Health Harmful Algal Bloom System. And you can go to their webpage. I can drop it in the chat later. And you can actually see like the states who report to this surveillance system. I believe there's like 13 states who report. You can see data about illnesses, how many are reported, how many blooms are reported. You can look at like demographic information, all sorts of variables associated with these reports. So I highly recommend checking out that website. Again, I'll drop it in the chat later, but yeah, you can use that to kind of compare across states.
Yeah, that's great. Thank you. The next question is by Wanda. How many samples are typically taken for possible HABs toxins? And are other toxins analyzed beside MCLR?
So we usually collect samples in response to an illness. The DNR staff will collect either 1 or 2 samples, and then they're sent to our state lab, and then it depends on what sort of testing we'll do to know which type of toxin that we want to look at. So sometimes we won't even like test for toxins. We'll only test for like cyanobacterial species, but we'll do that species testing first. And then based on those results, if there's like a likelihood that toxins may be present, we'll then do toxin testing. And Gina might be able to elaborate on that in the chat. I might have totally like butchered that process, but I don't know if that answered the question on like how many samples we take. It's usually just 1 sample from the exposure site. But Gina,
Thank you.
Can elaborate, that'd be great.
Yeah, the next question is by Jim Olson. What type of communication did you find was most effective? Was the information received well by the community? And yeah, we try to say, okay, this I guess we try to say that this water has some problem, HAPS problem. We communicate with the community.
Is this in reference to the illness outbreak?
I think so.
Okay. From my understanding, it was well received. At the state, we're sort of removed from that community outreach aspects. What we really do is we give the local health departments the tools and education needed so then they can educate the people in their community. So to an extent, I'm not sure how well or how much this information was received from that standpoint, but I imagine pretty well. I would, or at least I hope so. I know hearing back from the local health departments, people were pretty receptive to seeing those flyers and receiving them. And it's interesting because in that particular scenario, that beach was a beach that was not known for experiencing HAB. So it was kind of this whole big, a pretty big deal because, you know, the beachgoers who go there annually and the camp staff and the local health departments are not familiar with this issue. So it was definitely a learning curve trying to educate the local health departments who could then educate their community members. But yeah, in short, I assume that the information was received well.
Yeah, I actually is also thinking about this. You mentioned that sometimes it's very difficult to make a direct connection between illness and HAPS because it's difficult. Some people, whether they would say that, well, just try to be dismissive. I really just say that the direct connection may not be there.
Mm-hmm. Yeah, it's tough. And with that, you know, we can receive these surveys. We receive the illness complaints, we follow up. Sometimes we can't even reach the illness complainant to get more information. So that also like presents a challenge in itself, is just making that initial contact with the illness complainant to learn more. But it is honestly can be pretty challenging to determine whether an illness is related to bloom exposure. One thing we really rely on when people report illnesses to us are photos of the water conditions when they were recreating. Someone could be recreating in what they describe as green water, but it could in the end just be normal filamentous algae and not toxic algae. So we always encourage people to submit photos for us because that helps immensely with the investigation process.
Okay. Yeah, there are a few more questions there on the Q&A box there, but the time is running out. So if you could get around, probably you could just go there and respond to those questions there. Okay.
Sure. Yeah, I can do that.
Yeah. I just want to thank everyone for coming to this session, for this annual Harmful Algal Bloom Symposium there. Just a reminder there, the recordings and PowerPoint slides will be posted to our website at northcentralwater.org/HAPS. So please take a look if you want to get into sort of more details on that, want to review some of those presentations and the slides again. And also, if you have any questions there, this screen shows the email address for Lindy Holt and Jordan Murray. You can reach out to them about specific questions for that. With that, I and others, do you have any questions before we close the symposium?
No, I don't think so. Thanks so much, everyone, for your time. Thank you to our speakers and to the facilitators.
Thank you. Hope everyone have a good day. See you next time.